Hey everyone, welcome to the PowerPoint recording for the cell part 2. We do still have two things about the cell that we need to talk about: cellular replication and genetics. So let's get started with cellular replication. So I'm sure you know, even in healthy people, there are always cells that are dying, right? But it's okay because cellular replication is also an ongoing process. So the cells that die are replaced by new cells. So there are two steps in cellular replication: proliferation and differentiation. What's the difference between those two? Basically, they are just what they sound like. Proliferation means the cells are proliferating or increasing in number. And differentiation means the cells that have been newly formed are becoming different from each other. They're specializing. They're developing some unique capabilities. So this slide might give you a better idea of what cellular replication is about. At the top, you see proliferation, where new cells are being formed. And then you see all the different levels of differentiation that those new cells have to go through before they end up as the mature cells that you see along the bottom. And if you look at what those mature cells are, you know that those cells all do have some unique characteristics. Proliferation can occur either by mitosis or meiosis. Do you remember what the difference between those two is? Mitosis is the process through which your regular body cells replicate. Sometimes we call those regular body cells somatic cells. Meiosis is the process through which the germ cells reproduce. And the germ cells, you remember, are the cells that go on to become the egg or the sperm. So bottom line, germ cells reproduce by meiosis. All the other cells in your body reproduce by mitosis. Let's talk about mitosis first. So you remember in mitosis that you have a parent cell that divides into two daughter cells. And remember, each of the two daughter cells is supposed to have the exact same set of chromosomes that the parent cell had. And since we're talking about mitosis, that means 46 chromosomes, right? The parent cell has 46 chromosomes, so each of the two daughter cells should have 46 chromosomes. Now, what is it that determines how often a cell is going to proliferate? There are basically three things that can affect proliferation, the most important of which is genetic programming. It's genetically pre-programmed into each type of cell how often it should divide. Different kinds of cells reproduce at different rates. For example, liver cells normally divide only once every 1 or 2 years. Whereas the epithelial cells that line your GI tract, those might divide maybe 2 or more times a day. So it is genetically programmed into each type of cell how often they should divide. That's the primary influence. There are a few other things that can affect proliferation, though. For example, we have what we call cell density dependent inhibition. And that sounds like a big fancy word, but all it means is that cells are going to proliferate until they start coming into contact with a whole bunch of other cells, and they start to feel crowded. And so then proliferation is going to slow down. For example, let's say you have a big wound in your arm, and the cells are going to proliferate to try and fill in that wound and heal it over. When the edges of that wound start coming in contact with each other and the cells start feeling a little crowded, that's sort of the signal that tells proliferation to slow down, because the wound is healed over and we don't need that much proliferation anymore. Then there are some situational factors that can affect proliferation. Things like the amount of growth factor that's present, the amount of hormones or nutrients that are available, or maybe events like blood loss, tissue injury. Those kinds of things can signal that proliferation needs to occur. But again, the primary thing that affects proliferation is what's programmed into that cell's genes. So this is a diagram of the cell cycle, which is the process that the cell goes through as it proliferates. And you can see it's divided into 4 main phases. In the G1 phase, the cell's getting ready to divide, right? So it needs to make more proteins and RNA and more of those little organelles, or those structures that we looked at last class period that are in the cytoplasm of the cell. Because after all, if the parent cell is going to divide into 2 daughter cells, it needs to have enough RNA and organelles and all those kinds of things to fully supply 2 new cells, right? The next phase is the S phase, and S stands for synthesis. And what we're synthesizing in the S phase is DNA, because again, if the parent cell is going to split into 2 daughter cells, it needs to have enough DNA to supply 2 cells. The G2 phase is pretty much a repeat of the G1 phase with similar kinds of things going on. And then the M phase, M stands for mitosis, and this is the phase in which the cell actually splits into 2.
You might want to say, wait a minute, did you say 4 phases of the cell cycle? Because it looks like there's 5, right? What's this G0 thing? Okay, well, G0 is considered the resting phase. Most people don't consider G0 to be part of the cell cycle. They consider G0 to be a time where the cell is out of the cell cycle. It's resting. Okay, when we say it's resting, we're talking in terms of cellular replication. Okay, the cell is not doing anything in terms of proliferation. Okay, of course, we don't mean that the cell is doing nothing, right? Because the cell does have to do all those things that it needs to do in order to stay alive, like make ATP and that kind of thing. But it is resting in terms of cellular replication. The cell is not getting ready to divide. It is not dividing. It is just resting. Now, of course, there is a cell cycle sort of like this in meiosis as well. The exact things that happen in the phases are a little different in meiosis than they are in mitosis. But I'm not going to burden you with that at this point. I just wanted you to be familiar with the general concept of a cell cycle. But in terms of the difference between meiosis and mitosis, remember we said at the end of mitosis, each daughter cell ends up with the exact same set of chromosomes as the parent cell. And we noticed that it was 46. Okay? Remember, the big difference when you have meiosis is at the end of meiosis, instead of each of the new daughter cells having 46 chromosomes, how many chromosomes do they have? Right, they only have 23. Not 23 pairs like in mitosis, but just 23. Now, why would that be? Okay, remember we said meiosis is the process through which the germ cells replicate. And remember, each of the egg and sperm cells only have 23 chromosomes, because when the egg and the sperm get together to make the zygote, then between the two, you will have 46 chromosomes or 23 pairs. If each of the germ cells started out with 46 chromosomes, when they got together, then the zygote would just have way too many chromosomes. Okay, so big difference between meiosis and mitosis is the number of chromosomes that you get in the new cells that are made. Okay, so maybe you're thinking, why do I care about the cell cycle? Okay, let me tell you why you care. When we get around to talking about cancer, the cell cycle will help us to understand a little bit more about how cancer develops as well as how we treat it. In cancer, we're going to talk about how there are a higher number of cells actively in the cell cycle replicating and fewer cells in the resting phase than there should be. And that's what leads to that uncontrolled cell growth that we see in cancers. We have tumors that are getting bigger, growing out of control, and so forth, because there's more cells actively dividing. Then when we get around to talking about treatment for cancer, and we talk about the different kinds of drugs we use for chemotherapy, you're going to learn that a lot of the chemotherapeutic drugs are what we call cell cycle specific, meaning that some of those drugs work specifically during one of these phases of the cell cycle to interfere with what's supposed to happen during that phase and therefore keep the cancer cell from replicating. So some drugs are active in the S phase. They prevent the DNA from being replicated, maybe by interfering with enzymes that are needed for that particular process. Other drugs may work specifically in the M phase to keep the cancer cell from actually dividing into two. If you understand that about how the chemotherapeutic drugs work, then it helps us also to understand a little bit more about how we prescribe these drugs. For example, it explains why we often put people on more than one chemotherapy drug at a time, because we might pick one that works during the S phase and one that picks during the M phase, and as we do that, then we're assured that we'll probably be able to wipe out more cancer cells than we would if we only used one particular kind of drug. It also explains why very often we have to give chemotherapy drugs for a fairly extended period of time, because if we're giving a drug that works only during one phase of the cell cycle, then what that means is we have to give the drug long enough for hopefully all of the cancer cells to eventually cycle through to that phase of the cell cycle before we can halt its replication. Now, understanding the cell cycle will also help us when we get around to talking about genetic disorders, because if something goes wrong during the S phase and the DNA gets replicated wrong, then that can lead to certain kinds of genetic disorders. Also, we sometimes see in the M phase, when the parent cell splits into the two daughter cells, the chromosomes don't get divided up equally, and that leads to a lot of chromosomal disorders, and we'll talk about those a little bit more in class.
So all that stuff we've been talking about has to do with proliferation. But remember, there are two steps to cellular replication, and the second step is differentiation, where the new cells are developing into different, more specialized kinds of cells. So let's talk a little bit about differentiation now. You have in your body cells of varying levels of differentiation. You have some cells that are very highly differentiated, highly specialized, like your nerve cells or your skeletal muscle cells, cells that can do things that other cells cannot, like conduct action potentials. The thing that we've noticed about these highly specialized cells, though, is that they are much less able to reproduce. You remember in our last recording, we talked about your brain cells, which are highly specialized, but for a long time we thought that brain cells could not reproduce at all. We now know that they do reproduce, but they do it at a much, much slower rate. So it does seem that the cells that are highly differentiated are less capable of reproducing. Next, we have the parent cells, or the progenitor cells. These are cells that are kind of at a mid-level of differentiation. Cells like your skin cells or your liver cells that maybe can't do as many fancy things as the highly specialized cells can, but they are much more capable of reproducing. Your skin cells reproduce a lot, very often. But they are not as specialized. They can't do things like conduct action potentials. And yes, they are more capable of reproducing, but when they reproduce, they really only produce other cells that are just like them. So they can really only produce that one type of cell. Finally, we have some cells that are fairly undifferentiated. These are cells that don't have many specialized capabilities, but they are able to enter into the cell cycle and produce large numbers of new cells when they need to. And we know that these very undifferentiated cells can usually produce more than just one kind of cell.
Okay, so here you see the stem cell in your bone marrow. when it reproduces, it doesn't just make another stem cell, but it can differentiate out into all of the blood cells that you have in your blood. So we not just have red blood cells or platelets, but all of those different kinds of white blood cells that you see there can all come from the stem cell. So the undifferentiated stem cell can't do all the fancy things that maybe a nerve cell can do, but it is really good at replicating and creating not just one kind of cell, but several kinds of cells. So what you can see from this is that a very differentiated cell is not really more valuable than the undifferentiated stem cell. You need both kinds, right? You need the cells that are very specialized, and you need the cells that are very capable of reproducing. Okay, so what kinds of things affect how differentiated a cell becomes? Again, the most important thing that affects how differentiated or how specialized a cell becomes is genetic programming. What's genetically pre-programmed into each cell. Now, if you're thinking, maybe it's occurring to you right about now, wait a minute, didn't we just say that all of the cells in your body come from that original zygote, right? And when the parent cell divides into the two daughter cells, each of those two daughter cells is supposed to have the exact same set of genes and chromosomes and DNA that the parent cell had, right? So if that's true, then every cell in your body should have the same set of genes and chromosomes. So how can we say that genetic programming is the thing that determines which cells are going to become very specialized and which ones aren't? Okay, so it is true that every cell in your body pretty much has the same set of genes and chromosomes. But in every cell, there are some genes that are, quote, switched on and some that are, quote, switched off. Okay, so depending on which genes are switched on or switched off inside each cell, that's what's going to determine how often the cell proliferates and how differentiated it's going to become. Okay, so genetic programming, which genes are being expressed or, quote, switched on in the cell, is the primary thing that determines how specialized that cell becomes. Of course, there are some situational factors that will affect differentiation as well. Things like growth factors and nutrients that are present, how much oxygen there is, whether the cells get crowded or not. So some of the similar things that affect proliferation will also affect differentiation. But in both processes, remember the primary thing that determines what's going to happen with that cell is what's genetically programmed into that cell.
So, probably the most well-known problem that occurs when something goes wrong with cellular replication is cancer. Cancer is a disorder in which both steps of cellular replication are messed up. So both proliferation and differentiation are abnormal in cancer. Cancer is not really a single disease. I'm sure you know there are many kinds of cancer, skin cancer, lung cancer, bone cancer. And each of those kinds of cancers has some characteristics that are unique, but there are certain things that pretty much all cancers have in common, and those are the things that we're going to talk about today. As you progress through the curriculum, you'll probably study about specific types of cancers, but for today, we're just going to talk about cancer in general and the things that almost all cancers have in common. Now, your book defines cancer as a condition in which tissue growth exceeds that of normal. It's not appropriate, serves no useful purpose. In fact, it's even hurting the host, and yet it's continuing. So we have cancer cells that are growing out of control, that is not helping, it is even hurting people, but it's continuing anyway. That's what we describe as a cancer. And at the current time, we believe cancer cells are a result of genetic mutations. So, what is a mutation? Do you remember? A mutation is a change, right? So if we're talking about a genetic mutation, we're talking about a change in the person's genes. And if you think about it, that should make sense, right? Because we just got through saying that proliferation and differentiation are determined by genetic programming. So it makes sense, doesn't it, that if you change those genes because you have a mutation, now we're going to change the programming and we're going to have abnormal proliferation, abnormal differentiation, which is our definition of cancer. Does that make sense? So, if you were to look at a list of risk factors for cancer, you would probably see things that are capable of causing genetic mutations, like exposure to chemical or environmental carcinogens. Now, carcinogen is just a fancy word that means something that can cause cancer. But if you look at a list of things that are considered to be carcinogens, you're going to see things like exposure to radiation, even radiation from the sun, exposure to tobacco smoke, asbestos, toxic chemicals. These are all things that we learned from our last recording are capable of generating free radicals. And you remember free radicals are substances that go and steal electrons from your cell parts and cause cell damage. So if you have somebody who's exposed to a free radical that steals electrons from their genes and from their DNA, that's causing a genetic mutation. And depending on what the mutation is, it is something that can predispose to cancer. Does that make sense? Now, a lot of times there's a pretty big time lag between when somebody's exposed to the carcinogen and when they actually get diagnosed with cancer. I remember when I was 10 years old, one of my uncles who had been smoking for almost 2 decades decided to quit. And I remember that because he really had a hard time, but he was successful. And that was when I was 10. When I turned 40 was when he was diagnosed with lung cancer. So you see, there was like a 30-year time lag. Time lag can be anywhere between 5 to 30 years between when the person's exposed to the carcinogen and when they actually are diagnosed with the cancer. And that time lag can sometimes make it difficult to track back what the risk factor was. But usually if you look and you know the kinds of things to look for, you can pretty much identify the risk factors. Does that make sense? Another risk factor for cancer is exposure to cancer-causing viruses. Now, not all viruses will predispose you to cancer. So the next time you get a cold or the flu, you don't have to worry that you're at increased risk for cancer. But there are some viruses that are associated with certain kinds of cancers. Like, I'm sure you've heard of the link between HPV, human papillomavirus, and cervical cancer in women. There's also a link between the hepatitis B and hepatitis C virus and liver cancer, or the Epstein-Barr virus and certain kinds of lymphoma. So certain kinds of viruses can predispose to cancer. And if you think back to what you learned about how viruses replicate, that should make sense. So you see in this diagram, kind of like a life cycle of a virus, right? You remember that viruses are not like bacteria. Bacteria can replicate all on their own outside your body cells. But viruses can't do that. Viruses have to go into your body cells, and they use your body cells' machinery to replicate. So in picture number 1, you see the virus entering a host cell. In picture number 2, it's taking its viral DNA and incorporating it into the host cell's DNA. And in picture number 3, you see that that host cell now has DNA that's kind of like a combination of its own DNA plus the virus's DNA. Now, you might remember from our last recording that DNA has the instructions for how to make protein, right? So now that the virus's DNA is incorporated into the host cell's DNA, what is that host cell now going to make? It's going to make more copies of the virus, and that's how the virus replicates. Okay, but the point here being, when that virus sticks its viral DNA in the host cell's DNA, that's a genetic mutation, right? That has changed the host cell's genes, and now that the genes are different, that could lead to abnormal proliferation, abnormal differentiation, and therefore to cancer. Does that make sense? A third risk factor for cancer is immune system defects. There is a hypothesis called the immune surveillance hypothesis that says that probably cancer cells are developing in everybody's body all the time. It's just that our immune systems are supposed to recognize that those cells are abnormal and get rid of them before they have a chance to establish and grow and cause problems. So according to this theory, if there's a problem with your immune system and it's not working correctly, then it's not going to recognize the cancer cells or get rid of them. There is some support for this hypothesis because we do notice that people with immunodeficiency disorders like AIDS or people who are on immunosuppressive drugs, those people all have a higher incidence of cancer. Even old people have a higher incidence of cancer. And that makes sense because, unfortunately, as we get old, everything in our body gets less efficient, including our immune system. And so we kind of expect older people to have a weaker immune system and therefore be at higher risk for cancer. Does that make sense? The last risk factor I'm going to mention is heredity. If a cancer-causing genetic mutation occurs in a germ cell, in other words, an egg or a sperm cell, and if that egg or sperm went on to become your child would inherit that genetic mutation and would be at increased risk for cancer. Now, if you had a genetic mutation in one of your regular body cells, you might have a problem, but you wouldn't pass that on to your kids. If that genetic mutation occurs in a germ cell, though, that does get passed to your kids, and they would have a hereditary predisposition to cancer. I'm sure you've heard of certain kinds of cancers that are highly hereditary. I'm thinking of a particular kind of breast cancer that is so highly hereditary that women who carry that genetic mutation often have preventive mastectomies, so they don't have to worry about when am I going to get breast cancer. So there is a hereditary element to certain kinds of cancers. Now, because cancer is not a single disease, it is possible that different kinds of cancers are more related to different ones of these risk factors. But the bottom line is that most cancers probably are not due to just one of these things, but to a combination of these things. So the more of these risk factors a person has, the more likely they are to get cancer. Does that make sense? Okay, let's talk next about what kinds of problems cancer can cause for people. We said that cancer is a problem where both proliferation and differentiation are abnormal. So let's see what that would mean, starting with abnormal proliferation. We've already said that in cancer, there's a higher percentage of cells that are actively in the cell cycle proliferating than there usually is. We have fewer cells that are resting than usual. We also notice that cancer cells seem to lose their cell density dependent inhibition. They don't really care if they get crowded. And we find that cancer cells tend not to die within their normal genetically pre-programmed time frame. So we have cells that are proliferating faster than normal. They don't care if they get crowded, and they don't die when they're supposed to. That all leads to uncontrolled cell growth. Now, why do you think this is a bad thing? Why would it not be a good thing to have a whole bunch of extra cells around just in case you need them? The reason that uncontrolled proliferation is bad is because the cancer cells will increase in number to the point where they start to crowd out the normal cells around them. They steal the blood supply and therefore the oxygen and the nutrients from the normal cells. And the normal cells get to the point where they can't function. What about abnormal differentiation? What kinds of problems is that going to cause? Cancer cells don't differentiate or specialize properly, so they don't really do what the normal cells are supposed to do. They don't really act like the cell that they're supposed to be. So a liver cancer cell will not do the things that a normal liver cell would do. A renal cancer cell would not do the things that renal cells are supposed to do. So one of the things that you expect to see when somebody has cancer is dysfunction of whatever tissue or organ has developed the cancer. So in other words, if somebody had liver cancer, their liver wouldn't be able to do what it's supposed to do. We'd be looking at somebody eventually going into liver failure. And the same with renal cancer. Eventually we'd expect that person to go into renal failure. Also, because cancer cells don't properly differentiate, they sometimes have some different characteristics than normal cells would. For example, normal cells have a kind of cell-to-cell cohesiveness where they like to stick together. Cancer cells lose that cell-to-cell cohesiveness, so they don't mind if they break apart from their buddies and wander off to other parts of the body. So cancer cells frequently spread through the blood or through the lymph to other parts of the body and start cancers there. We call that metastasis. Because cancer cells don't function normally, they also sometimes make some weird substances that normal cells don't. For example, some cancer cells make a substance called vascular endothelial cell growth factor, or VEGF. Now that seems like a big long name that doesn't make any sense, but if you break it down, it actually does. Vascular means blood vessel, right? Endothelial cells are the cells that line the blood vessels, and a growth factor is something that promotes growth, right? So VEGF is a substance that promotes the growth of new blood vessels. This helps the cancer cell because normally a tumor would only be able to get to a certain size before it's not getting enough blood and therefore not enough oxygen and nutrients to get any bigger. If that tumor, however, were able to grow new blood vessels and to keep increasing its blood supply, that tumor could continue to grow. So VEGF is a substance that some cancer cells produce that actually helps the cancer continue to grow and get bigger. Other weird substances that some cancer cells produce because they didn't properly differentiate are ectopic hormones. Now you know what a hormone is, so what does the word ectopic mean? Ectopic means in an abnormal location. Like, I'm sure you've all heard of ectopic pregnancies, right? What's an ectopic pregnancy? It's a pregnancy that is not occurring in its normal location. It's not in the uterus where it's supposed to be. Maybe the embryo is in the fallopian tube or something like that. So the word ectopic means in an abnormal location, and ectopic hormones are hormones that are not produced by the endocrine glands like they're supposed to be, but they're being produced by the cancer cells. Now, if the cancer cells are producing hormones that are just like the hormones that your endocrine glands make. Why do you think this is a problem? Because remember, your endocrine glands are regulated by a negative feedback mechanism to make sure they don't overproduce hormone. The cancer cells are not regulated by any feedback mechanism, and they don't care if you already have a ton of hormone. They're just doing their thing, making ectopic hormones, regardless of how much you already have. So when cancer cells make ectopic hormones, we end up looking like we have endocrine diseases. We have hormone imbalances, usually too much. Okay, so an example of an ectopic hormone production would be a lot of lung cancers will produce hormones that are just like ACTH or antidiuretic hormone or a parathyroid hormone. And since those lung cancer cells are not regulated by that feedback mechanism, they're overproducing those hormones, and so those cancer patients look like they have endocrine diseases. Does that make sense? The last weird substance I'm going to mention that some cancer cells produce, some of them make enzymes or toxins that can actually damage cells. And unfortunately, tissue that's damaged by these enzymes or toxins often doesn't heal normally. So if you look at a lot of these lists of warning signs of cancer cells that you find in different public service announcements, a lot of times they list a sore that doesn't heal. Because remember, the damage that's done by these enzymes or toxins often doesn't heal normally. Sores that don't heal normally are often different kinds of skin cancer cells. And here you can see some examples of sores that didn't heal. One last thing that I want to mention is that many cancer cells, because they didn't differentiate properly, have some abnormal antigens on their cell membranes. And because we know this, we sometimes use those abnormal antigens as clinical markers to help us monitor the progression of a cancer or to evaluate the success of treatment. Here are some examples of abnormal cancer antigens. You might have heard of PSA, which stands for prostate-specific antigen that we find on the cell membrane of some types of prostate cancer cells. We also have CEA, which stands for carcinoembryonic antigen. We find that antigen on certain kinds of GI cancer cells, like colon cancer, gastric cancer, pancreatic cancer cells. Also, we might find it on certain kinds of breast cancer cells. Now, these antigens are not used to diagnose people with cancer or to screen people for cancer because they're not really reliable for that purpose. Some of these antigens might appear in some non-cancerous conditions, and sometimes they don't appear until cancers are fairly advanced. And, we'd really like to find the cancer before it gets advanced. So it's really not used to diagnose or to screen for cancer. How we use it, though, is usually to help monitor the progression of a cancer or to evaluate how successful treatment of cancer might be. Let me give you some examples. Let's say we have a guy that we know has prostate cancer. We've done a biopsy and we've already diagnosed the prostate cancer. What we would do is we would probably draw his blood and measure PSA levels, and then we would pick a chemotherapy drug and start the guy on it. After a certain amount of time goes by, we will draw blood again and recheck PSA levels. And let's say we find that the PSA levels are still high or maybe have even gotten higher. What are we going to think? We're going to think that that drug isn't working, right? So we're going to switch to a different drug. and leave the patient on it for a while, and then draw blood again and recheck PSA levels. And let's say after we change the drug, we check PSA levels and they're dropping. Now what are we going to think? Yeah, we're going to think this drug is working. Let's leave him on it. Okay, so we finish the round of chemo and, we get to the point where we decide this patient is in remission. Once we get to that point, we will periodically ask the patient to come in and have his blood drawn, maybe like every 6 months for the first couple of years. And, let's say maybe 5 years down the road, we have the guy come in and we see the PSA levels are starting to go up again. Now what are we going to think? Yeah, we're going to think maybe the cancer's come back. We ought to at least get this guy in and check it out. So does that make sense how we use it? We don't use those antigens to diagnose or to screen, but we do use it to monitor the progression of a cancer. Has it recurred? Or we use it to evaluate how successful treatment is. So now that we've talked about the problems that cancer causes due to abnormal proliferation and differentiation, let's talk about some more generalized effects. Problems that can happen for a combination of reasons, like cachexia. Do you know what cachexia means? Have you had this in medical terminology yet? Cachexia means weight loss, where people just kind of wasting away. So here's a picture of a patient with cachexia, and do you see how skinny he is? If you've ever seen a patient with advanced cancer, aren't they usually really skinny like this? There's a couple of different reasons why they lose so much weight. First of all, you remember we said that cancer cells steal the blood supply and the nutrients from the normal cells around them. So the cancer cells grow, but the patient gets really skinny. A second reason why the patients might lose so much weight is because the immune system, remember the immune system is supposed to be trying to get rid of cancer cells. And one of the ways the immune system does this is by releasing cytokines. Do you remember what cytokines are? We'll talk a lot more about them in a couple weeks when we talk about the immune system. But for now, let's just define a cytokine as a substance that white blood cells make to help them do their job. Okay, so there are substances that white blood cells make to help them do their job. And remember we said part of the immune system's job is to get rid of cancer cells. And one of the ways it does that is by releasing cytokines that are supposed to kill the cancer cells. Unfortunately, cytokines often have the side effect of causing anorexia. Anorexia means loss of appetite. So cachexia means weight loss. Anorexia means loss of appetite. And, since the cytokines cause anorexia, people lose their appetite. They don't want to eat. Of course they're going to lose weight, right? Have you ever seen these advanced stage cancer patients that have lost a lot of weight and their families come in to visit them and they bring food that they cook them from home? I made your favorite food. Please eat it. You're getting too skinny. You have to eat. The patients are just like, I can't eat it. I'm just not hungry. That's the anorexia that's caused by the release of cytokines. Does that make sense? Another common problem that people with cancer have is pain. I've had a lot of people with advanced cancer tell me stuff like, I'm not afraid to die. I just don't want to die in a lot of pain. Pain might be one of the more challenging aspects of cancer that you'll deal with as nurses. Because a lot of times in order to adequately control a person's pain, we have to give enough pain medication that they become sedated. And people who are in the end stages of cancer don't want to sleep away the time that they have left. They want to spend it with their families. So it can be a real challenge to manage people's pain. There are a number of reasons why people with cancer might have pain. Sometimes as tumors grow, they start pressing on nerve endings, and that triggers pain. Or as tumors grow, they might press on blood vessels in the tissues around them and kind of cut off blood flow. So when tissues aren't getting enough blood and oxygen, sometimes that triggers pain. We also mentioned earlier that some cancer cells make enzymes or toxins that damage tissue, right? Well, damaged tissue hurts. So that's another reason why pain might happen. There are some experts that think that there's something about cancer that directly causes the release of some of the chemical mediators of pain. So again, a lot of reasons why people with cancer might have pain. Finally, a lot of cancer patients have problems with anemia. Do you know what anemia is? Yeah, anemia is when there are lower than normal levels of red blood cells or hemoglobin. And there's a lot of reasons why cancer patients might become anemic. First of all, remember we said that cancer causes anorexia, right? And so people might develop anemia because of poor nutrition, nutritional deficiencies like iron deficiency or B vitamin deficiencies. We also said that sometimes cancer cells make toxins that damage tissue. And damaged tissue often bleeds. So the patient might become anemic because they're losing blood. Or sometimes as tumors grow, they erode into blood vessels. And now you have a much bigger blood loss that can cause anemia. Sometimes cancers metastasize to the bone marrow. And, if there's cancer of the bone marrow isn't going to be able to make red blood cells, so people become anemic. Ironically, sometimes cancer patients get anemic not because of the cancer, but because of the treatments that we're doing to fight the cancer. Often we do chemo or we do radiation, and both of those treatments can suppress the bone marrow and therefore cause anemia. So people with cancer might become anemic because of the cancer itself, or they might become anemic because of side effects from the ways we're treating the cancer. So when you think about all the problems we've talked about, can you see why if we don't control the cancer, people are going to die, right? So this is probably a good time to differentiate between benign growths and malignant growths. If you had a tumor, would you rather have a benign one or a malignant one? Benign, right? Why? Because benign growths are not cancerous. Malignant growths are. So remember we said cancer involves problems with both steps of cellular replication. Both proliferation and differentiation are abnormal in cancer. In a benign growth, only proliferation is abnormal. Differentiation is totally normal in a benign growth. So what that means is people with benign growths are really only going to have like half the problems as people with cancer do, right? They only have problems because the tumor is getting bigger. They aren't having problems because the cells aren't working properly. Okay, so a benign growth is going to get bigger by expansion. Just as the cells proliferate, the tumor is going to get bigger. Whereas the cancerous growths, because they aren't acting normally, they might grow by expansion, but they'll also spread by metastasis. Remember, because the cancer cell didn't differentiate properly, they don't have that normal cell-to-cell cohesiveness. They don't mind breaking apart from their friends and traveling to other parts of the body. So cancerous growth spread by metastasis, and they start infiltrating into surrounding tissues. Whereas benign growths not only are just growing by expansion, but very frequently benign growths are surrounded by a fibrous capsule that the cancerous growths don't have. And so, do you see how when there's a capsule around the benign growth, it's a lot easier to surgically remove that tumor and to feel confident that you got all of it out. Whereas the cancerous growth doesn't have that capsule around it. And so we could try to surgically remove it, but it's so much harder to be confident that we got every cancer cell out. And you know, with the cancer cells, if you even leave a few of them behind, what's going to happen? Yeah, they're going to keep multiplying and keep growing, and the cancer is going to come back. So here we have a picture of a cancerous growth. The cancer is the lighter colored part in the middle, and the yellowish tissue is the normal tissue. So you see how the cancer is sending out little tentacles that is infiltrating the normal tissue. It's not surrounded by a capsule, and so you can imagine how difficult it would be to try to surgically remove that and feel like you got it all out. This, on the other hand, is a benign growth. The tumor is, again, the lighter colored part, and the reddish part is the normal tissue. And you can see how there is a capsule around this benign growth that's keeping it contained, and it's keeping it from infiltrating into the surrounding tissue. So obviously it'd be a lot easier to surgically remove this tumor and to really feel confident that you got it all out. Other differences between benign and malignant growth include the fact that benign growths, because they are properly differentiated, they're going to work more like normal cells. And so they're not going to make all those weird substances that the cancer cells make. They're not going to make ectopic hormone. They're not going to make VEGF. They're not going to make those toxins and enzymes that damage tissue. They're not going to cause all those generalized effects of cancer like anemia or cachexia. Really, they're only going to cause problems in that local area that just surrounds the tumor. In fact, benign growths usually aren't going to kill people unless because of where they're located or because of their size, maybe they might interfere with some very vital function that you need in order to survive. For example, there are certain kinds of brain tumors that are benign but can still kill people because of where they're located. When they get too big, they might start squashing brain tissue and interfering with the brain's ability to regulate breathing or regulate heart rate or blood pressure. In those kinds of situations, you might see a benign growth actually kill somebody. But under most circumstances, benign growths don't cause the kinds of problems that cancerous growths do, and they are much less likely to cause death. Okay, let's briefly talk about some of the treatment strategies we use for cancer, just so we can see the logic behind why we use them. One of the more common treatment strategies is to surgically remove the cancer. And the logic behind that one is pretty clear, right? If we remove the cancer, We can remove the detrimental effects that cancer cells can have on the normal tissues around it. If the cancer cells are gone, they can't steal the nutrients and the blood supply from the normal cells and all of that kind of thing. However, we've already mentioned that it is hard to surgically remove a cancer and to feel confident that we got all the cancer cells out. And, if we leave even a few cancer cells behind, those are going to start to multiply and the cancer is going to come right back. For that reason, we almost always combine surgery with one of the other two common treatment strategies. The rationale for the use of radiation to treat cancer is pretty interesting. You remember that we said radiation is one of those things that can generate free radicals, right? So what we're doing when we do radiation is we're trying to generate free radicals that will either kill the cancer cell outright or will at least damage it to the point that it can't replicate. Of course, the downside to doing radiation is that some of the normal cells around the cancer might get radiated as well. But the thing that keeps radiation still a viable option is that it seems like the cells that are most affected by radiation are the cells that are most rapidly proliferating. And the cells that are most rapidly proliferating are usually the cancer cells. So that's what makes radiation still a good treatment option, even though it does cause some side effects. The last treatment strategy is chemotherapy, and we've already kind of talked about how chemo works, right? We said that a lot of chemo drugs work during a specific phase of the cell cycle to interfere with what's supposed to happen during that phase and therefore prevent the cancer cell from replicating. So some of the drugs are going to prevent the cell from making enzymes that it needs in order to function. Or it might prevent the cell from making DNA or making more protein. It might prevent the cancer cell from actually splitting into two. The downside to chemo, though, is most of the time these drugs are given by mouth or by IV, and so its effects are not necessarily just limited to the cancer cells. And that's the reason why chemo also almost always causes some side effects. Again, the thing that makes chemo a viable option is that it does seem like the cells that are most affected by chemo are the cells that are most rapidly proliferating, and of course, those are the cancer cells. The thing that I think is really ironic about radiation and chemo as treatments for cancer, though, is, when you think back to that list of predisposing factors, things that we said can cause cancer, isn't radiation and exposure to toxic chemicals and drugs, aren't they on that list of predisposing factors? So sometimes the things that we're using to treat cancer can actually predispose to a different kind of cancer later on down the line. But if you think about it, what choice do we have, right? Because if we don't do chemo and radiation, the patient dies now. If we do it, we might be predisposing them to other kinds of cancer later on, but at least we've bought the patient some extra time, right? So most people, you explain the risks to them, but they still choose to do it. I think I would as well. I just stuck this slide in to make you aware of some of the newer drugs that we use to treat cancer. We call them biologic response modifiers or immunomodulators. And those sound like big words that make no sense, but if you break the terms down, they actually do make sense. A biologic response modifier is just a drug that modifies or changes some kind of biologic response that's occurring in the body. And mostly we're referring to immune responses. So you remember that your immune system is supposed to fight cancer, right? And so a biologic is just a drug that changes the way the immune system acts to make it more effective in getting rid of the cancer. An immunomodulator is a drug that modulates or changes the immune system in some way. And so basically it's the same thing as a biologic response modifier. So you'll hear a lot more about these kinds of drugs as you go through the curriculum, because these drugs can be designed to fight lots of different kinds of problems. Some examples of these drugs are things like monoclonal antibodies. A monoclonal antibody is an antibody that's man-made. It's an antibody that we've cloned and designed to target some specific substance that is causing a disease. So once we figure out what kind of substance is causing the problem in a disease, we can design an antibody to get rid of it, and hopefully the patient improves. Lots of different kinds of monoclonal antibodies out there to treat lots of different kinds of diseases. Synthetic cytokines are just man-made versions of cytokines, and you remember we said a cytokine was something that your white blood cells make to help them do their jobs. There are lots of different cytokines, and we can synthetically produce a lot of them in the lab. And again, this is a way to modify or change how the immune system responds to different diseases and hopefully make it more effective in, treating the disease. So you are going to hear about these kinds of drugs a lot as you go along, and I just wanted you to have a little information about them so that they don't scare you when you see them. Okay? Okay, let's switch gears now and talk a little bit about genetics. I think the first thing that we need to do is define a few of our terms, because up to this point we've been throwing around words like DNA, genes, and chromosomes, and using them like they mean exactly the same thing. And while they are very closely related, they aren't exactly the same thing. So let's start with DNA. DNA stands for deoxyribonucleic acid.
And you see in this little picture here that DNA is a complex molecule that stores genetic information. And you probably remember from biology class or something like that DNA is made up of chains of nucleotides. And nucleotides are compounds that contain one of 4 possible nitrogenous bases. And it's the bases that carry the genetic information, that form the genetic code.
Now in this picture, if you look at the strand of DNA that the arrow is pointing at, you can see that the different bases are represented by different letters of the alphabet. And so when you look at that strand of DNA, you can see which bases appear and in what order they appear and in what quantity they appear. And that's what the genetic code involves.
In this little slide, you can see that when the DNA is translated into RNA, which is then going to carry the genetic code out into the cytoplasm so the ribosomes can use it to make proteins, you see that when the code gets translated to the RNA, we look at it in groups of 3. We call it a triplet code. And each triplet is called a codon. And each codon represents one specific amino acid. Okay, so what you've got here is in the DNA, we've got the triplet code, which, one triplet after another, each triplet representing an amino acid. And you remember that proteins are made of amino acids, right? So if you get enough of these codons that represent amino acids and you stick them together, pretty soon you have the instructions for how to make a protein, right? Because the codons are going to tell you which amino acids to use to make a particular protein, in what order and in what quantity that you're going to use them. So when you put all the codons together, eventually what you end up with is the instructions for how to make one particular protein. And that's what a gene is. Okay, so here you see the DNA molecule has genes that are hooked end to end to end to end to make this one big DNA molecule. And each of those genes is the instructions for how to make a specific protein. Now, we used to think that one gene had the instructions for how to make one protein, but we now know that there are some proteins that are so big and complicated that it takes more than one gene to store the instructions for how to make that particular protein. But if in your head, when you're thinking about it, if you think that a gene is the instructions for how to make a particular protein, then you won't be too far off track. You take that big long DNA molecule and you start winding it up really, really tight around a little protein core, which you see where that little red arrow is pointing at, to the little protein core, and the DNA molecules winding itself around it. Okay, so you get the DNA molecule, you wind it up really tight around the protein core, and then you keep winding it and winding it and winding it. And you see in that process, eventually you're going to end up with a chromosome. So what a chromosome is a DNA molecule that's wound up really, really, really tight. And so that there are many, many, many genes in each chromosome. Now, how many chromosomes are there in each cell? Okay, remember, most body cells have 46 of these DNA containing chromosomes. And those 46 chromosomes, remember, they're arranged into pairs, like this. So we have 23 pairs of chromosomes. And of course, one chromosome of each pair came from your mom, and one came from your dad. So they are all paired up like that. And you remember that 22 of the 23 chromosomes are what we call autosomes, which means that it doesn't matter whether you're male or female, they're alike, okay? And then the 23rd pair is your sex chromosomes, which determine whether you're male or female. And you remember, of course, that the males are XY and the females are XX. So this is a genotype for a male because the sex chromosomes are XY. So if you take all of that genetic information, all of the genes that are in all those chromosomes, and you put it together, you get the genetic information for one unique person. Okay, right there in that slide, you see the genetic information for one particular person. So you remember all the genes are instructions for how to make a particular protein. So right there in those 23 pairs of chromosomes, you have the instructions for how to make all of the different proteins that your body has to make. So let's summarize all of that, what we just said, into a little nutshell. Okay, so you remember the DNA is a big long molecule that's made up of chains of nucleotides. And the nucleotides each have one particular nitrogenous base in them that's represented by those letters of the alphabet, the A, the C, the G, the T. Okay, those bases are arranged in groups of 3, which we call codons, and each group of 3 represents one particular amino acid. Now, since proteins are made up of amino acids, if you take enough codons that represent amino acids and put them together in a particular order, then that provides the instructions for how to make a particular protein. And that's what a gene is, right? The genes are instructions for how to make a protein. And then when you stick all the genes together end to end, You get a big long DNA molecule, and then you wind up the DNA molecule around little protein cores. You make a chromosome, and then you take 46 of those chromosomes and stick it together into a cell, and you have the genetic information for one person. Okay, let me give you a little analogy to help you kind of understand what that's all about. If you take a bunch of letters of the alphabet and put them together in a particular order, in a particular quantity, you can make words. And if you take a bunch of words and put them together in a certain order, then you can make sentences. And if you take a bunch of sentences and put them together, you get a paragraph. You get a bunch of paragraphs and put them together, you get a story. Okay, so just like that, if you take the bases and the nucleotides and put them together in groups of 3, that represents an amino acid, and we call that a codon. You get a bunch of codons and put them together. You get the instructions for how to make a protein, which is what we call a gene. And then if you put the genes together and stick them end to end, you get a big long DNA molecule, which you then wrap up really, really, really tight, and you get a chromosome. And then if you take 46 of those chromosomes and put together into a cell, you have the instructions for how to make one person. Okay, so do you see the DNA, the genes, the chromosomes, they're all genetic material, but it's just a different size chunk of genetic material. Okay, so that's why sometimes it seems like we're using them interchangeably, but they're just referring to different size pieces of genetic information. Okay, so let's talk a little bit more about the gene. We said that the gene was the instructions for how to make a particular protein. Okay, so one of its main jobs is to regulate protein synthesis, right? And if you think about it, from what we've talked about as far as cellular structure and function, you know that cells are made of proteins, and you know that most of the cellular functions are carried out by proteins like enzymes and those kinds of things. So bottom line, if you want to put it into a really tiny nutshell, the genes control the function of the cell, right? Because the genes have the instructions for how to make the cell parts and how to make the enzymes and other proteins that are needed for the cell to carry out its functions. So the genes, through its regulation of protein synthesis, basically control the functions of the cell. The other thing that we said that the genes do, though, is they determine the inheritance patterns of each person. It determines what traits are going to be passed through the genes from one generation to the next. And we're not just talking about physical traits like what color your hair is, what color your eyes are going to be, but we're also talking about biochemical and physiologic traits like how fast or slow your metabolic rate is going to be, or if you have the predisposition for certain kinds of diseases like cancer. Now, the basic patterns of inheritance were first proposed by that guy named Mendel. You remember studying about him in some class somewhere? He published his laws, right, Mendel's laws, somewhere around the neighborhood of 1865, which is about the time of the Civil War. And we still call them Mendel's laws today, and we study them basically the way he proposed them all those years ago. And if you recall, Mendel's laws go something like this. Okay, you get the egg and you get a sperm and you put them together and you get a zygote. And the zygote has 23 pairs of chromosomes, with one chromosome from each pair coming from the dad and the other one coming from the mom. Now, because the chromosomes are paired up like that, that means that there are 2 genes for every trait, right? You have 2 genes that determine what your eye color is going to be. One gene came from your mom, one gene came from your dad. Okay, you have 2 genes that determine what your hair color is going to be. One from your mom, one from your dad. Okay, so all of your genes are paired up. And we sometimes refer to the 2 members of a gene pair as alleles. Okay, now, in each allele, or in each gene pair, sometimes there's one gene that is more influential than the other in determining what that specific trait is going to be. We call the more influential trait the dominant one, and then the other one is said to be recessive. Okay, now, if you have one dominant gene and one recessive gene, which gene is going to be expressed? Okay, the dominant one, right? So if you have one gene for brown eyes and one gene for blue eyes, what color eyes are you going to end up with? Okay, you're going to end up with brown eyes because brown in terms of eye color is the dominant gene. Okay, so a couple more terms to be familiar with: homozygous and heterozygous. Okay, homozygous. What does homo mean? Homo means same, right? So in a homozygous pairing of genes, or a homozygous allele, both of the genes are the same. They're either both dominant or both recessive. So you see, you remember when we were diagramming out genetic patterns, we use an uppercase A for the dominant gene and a lowercase a for the recessive gene. So if you have two dominant genes or two recessive genes, that's a homozygous pairing. Okay? Now, heterozygous. Hetero means different, right? So in a heterozygous pairing, you have one of each. You have one dominant gene and one recessive gene. So they're not the same, they're different from each other. That's a heterozygous pairing. Now, notice that a dominant gene is going to express itself whether it's in a homozygous pairing or a heterozygous pairing, right? Because in this particular slide with the heterozygous pairing, you have one dominant gene, one recessive gene. The dominant gene is going to express itself, right? So the dominant gene expresses itself whether it's in a homozygous pairing or a heterozygous pairing. The recessive gene is only going to express itself in a homozygous pairing, right? So the recessive gene only expresses itself if there is no dominant gene present. So in order for a person to have blue eyes, they have to have two blue-eyed recessive genes. If there's one brown eye gene and one blue eye gene, then that person's going to have brown eyes. So the recessive gene only expresses itself in the homozygous pairing. Now, if you have a heterozygous person, okay, like let's say this is eye color again, and they have one gene for brown eyes and one gene for blue eyes, we said that this person himself is going to have brown eyes, right? But can this person pass on blue eyes to his children? Yes, he can, because he does have the blue eye gene, and maybe he passes that one on to his kids instead of the dominant brown eye gene. So a heterozygous person with brown eyes himself can pass on blue eyes to his children. What do we call that person? Right, he's a carrier, okay? So he doesn't have blue eyes himself, but he is the carrier of blue eye genes.
All right, there are two kinds of disorders that I want to talk about: gene disorders and chromosome disorders. Now, you might remember that genes and chromosomes are both genetic material, just different size chunks of them, right? The gene is the smaller piece, Each gene is the instructions for how to make a particular protein, whereas the chromosomes have a lot of genes in them, right? So as you might imagine, if you have a chromosome disorder, you're probably looking at a lot more different kinds of problems than if you had a gene disorder. Not necessarily more serious or more severe problems, just a bigger number of problems. Does that make sense? So let's start with the gene disorders. Gene disorders are a result of genetic mutations or a change in a person's genes. During meiosis, when the DNA is being replicated, a mistake happens and the DNA is not replicated accurately, and now we have a genetic mutation. We're not always sure why this happens. Sometimes it's because the person was exposed to radiation or to toxic chemicals or something else in the environment. But a lot of times we never know why the mutation occurs. It can just spontaneously occur. Your book refers to it as a, quote, metabolic accident. Now, if there's a genetic mutation in one of your regular body cells, we don't really consider that a gene disorder. It can cause problems, but we don't call it a gene disorder. Gene disorders are what we call it when there's a mutation in a germ cell. Because you remember, if the mutation's in a germ cell, then it can be passed on to the next generation. Those are the ones that we're going to focus on today. A single gene disorder is what happens when there's a mistake or a mutation in one gene in a germ cell, and then that mutation gets passed on to the next generation. Of course, it is possible to have a mistake in more than one gene at a time, but when that happens, the inheritance patterns are a lot more complicated. So we're just going to look at the single gene disorders today, the ones where there's only one gene, and all the genes in all the chromosomes, there's only one gene that has a mistake in it. We call that a single gene disorder. Now, since we said that a gene has the instructions for making a specific protein, as you might expect, when there's a mistake in that gene, then there's some protein somewhere that we're either not going to be able to make, or we're going to make it, but it's going to be defective and it's not going to work. Okay, so the assessment findings for that gene disorder are going to depend on what protein it is that that gene has the instructions for making. So there really is no one blanket statement that you can make about symptoms of all single gene disorders. The symptoms of single gene disorders might be really different from each other because it all depends on what particular protein that gene has the instructions for and how important that particular protein is. So some single gene disorders are very mild and don't bother people very much. Other single gene disorders can be fatal if the protein is crucial for normal cell function. The same is pretty much true for interventions. There really is no one blanket statement we can make that would be true for all single gene disorders, because the interventions are going to be geared towards treating the symptoms of that particular disorder. And since we said the symptoms of single gene disorders can be very different from each other, the interventions might be very different from each other as well. At this point, we don't really know how to go in and fix the genetic mutation. So again, there really is no one thing that we can say that we do for all single gene disorders. Interventions are going to depend on what problems that disease causes. Does that make sense? Single gene disorders actually follow pretty closely the Mendelian patterns of inheritance. There are 3 primary patterns, and which one you see is determined by whether the defective gene is located on a sex chromosome or an autosome, and by whether that defective gene is carrying a dominant trait or a recessive trait. Let's talk about the sex-linked disorders first. In sex-linked disorders, the defective gene is located on one of the sex chromosomes instead of on an autosome. The sex-linked disorders are actually less common than the autosomal disorders because if you think about it, there are 22 pairs of autosomes and only one pair of sex chromosomes. So odds are if there's going to be a mistake in a gene on a chromosome somewhere, it's probably going to be on one of the autosomes instead of a sex chromosome. Now, when there is a defective gene on a sex chromosome, usually it's on the X, not the Y. And again, I think the reason is because there are just way more X's out there than Y's. Everybody has an X chromosome, right? Females have 2 X's, males have 1 X, but everybody has an X, whereas only males have a Y, right? So again, odds are if there's going to be a mistake in a gene on a sex chromosome, it's probably going to be on the X instead of the Y. That's why sometimes you hear people refer to sex-linked disorders as X-linked disorders, since the defective gene is usually located on the X chromosome. Now, most sex-linked disorders have a recessive inheritance pattern, meaning the defective gene is carrying a recessive trait. I've never been able to find a reference that explains why, but it does lead to a very interesting pattern. So you remember that a recessive gene is only expressed when there is no dominant gene present. So if the recessive gene is defective, it will only be expressed if there is no dominant or normal gene present to override it. Because females have 2 X chromosomes, if there's a defective gene on one of the X's and that gene is recessive, it's not going to be expressed, right? Because there is a dominant normal gene present on the other X. So in a recessive disorder, females usually are not going to have the disease. They're not going to have the symptoms. They might be carriers, but they're not actually going to have the disease. So on this slide, I've used a red color to indicate which X has a defective gene. So if you look at the female has 1 defective gene, but she also has 1 normal gene. And remember, the defective gene is carrying a recessive trait. The normal gene is carrying a dominant trait. So the dominant gene is going to express itself. That female will not have the disease. Of course, she does have a defective X, and she could pass that defective X on to her kids. But that would make her a carrier, not somebody with the disease. So what about the males? Males only have 1 X, right? And if that X has a defective gene on it, there is not another X with a normal gene on it to override that defective one. So the males with a defective X end up having the disease. So the pattern that we're left with is the males have the disease, the females are carriers. Now, you might wonder, is there not a normal gene on the Y that could override the defective gene on the X? I hate to break this to you guys, but you know, the Y chromosome is a lot smaller than the X chromosome. And actually, the X has more than 1,000 additional genes that the Y does not have. So you see, it's quite possible that there is not a normal gene on the Y to override a defective gene on the X. So we're stuck with the pattern. Males usually have the disease. Females are usually carriers. So here's a picture of a patient with hemophilia A, which is the most common X-linked disorder. In hemophilia A, the defective gene is the gene that has the instructions for how to make factor VIII. Now, do you remember what factor VIII is? It's a clotting factor. So people with hemophilia A have a lot of trouble with bleeding. And in this picture, you can see some of the other problems that the bleeding might cause besides just bruising. In the picture on the left, the patient has bleeding into a muscle, and in the picture on the right, the patient has bleeding into a joint. So do you see how the joint is all swollen? The other two inheritance patterns in single gene disorders are autosomal dominant and autosomal recessive. And in both of those patterns, the defective gene is located on an autosome and not a sex chromosome. So it doesn't really matter whether you're male or female as far as who's going to get the disease. The difference between autosomal dominant and autosomal recessive is that in autosomal dominant disorders, the defective gene is carrying a dominant trait. In autosomal recessive, the defective gene is carrying a recessive trait. So let's talk about the autosomal dominant disorders first. We already know that the defective gene is located on an autosome and that it carries a dominant trait. So if the defective gene carries a dominant trait, we know that it's going to be expressed, right? So we know even if there's only one defective gene, this person is going to have symptoms of that disease. So are there any carriers in an autosomal dominant disorder? No, because all it takes is to have one defective gene and you have the disease. And by the way, a carrier is defined as somebody who can pass on a disease but does not have symptoms themselves. A person with one defective gene can pass the defective gene on, but also has symptoms themselves. So they are not considered carriers. They are considered somebody with disease. So if you look at the example below, you can see that the defective gene is the uppercase A, and all it takes is one to give the person a disease. So in order for somebody to not have an autosomal dominant disorder, they have to have two normal recessive genes. Does that make sense? So an example of an autosomal dominant disorder is Marfan syndrome. And in Marfan's, the defective gene has part of the instructions for making connective tissue. And you know there's connective tissue all over your body, right? So the symptoms in Marfan syndrome span multiple body systems. We might see musculoskeletal problems, like one of the things that you see is really long, thin arms and legs. So this diagram is kind of showing you the arm span of somebody with Marfan's, which doesn't look that bad in this picture, but let me show you another one. In this picture, it's much more obvious, right? How long this person's arms are. You see the long, thin extremities, and also they often have chest and spinal deformities. So he looks like he's got some scoliosis and maybe a little hunchback kind of thing going on as well. And in this picture, you can see the long, thin fingers too. But remember, there's connective tissue all over the place. So in addition to the musculoskeletal deformities, there might be eye problems. For example, the retinas become detached or the lenses get dislocated because the ligaments are weak. The connective tissue in the ligaments is weak. We also tend to see a lot of cardiovascular problems. Some of the heart valves prolapse, there tends to be aneurysms and those kinds of things. What about the autosomal recessive disorders? Okay, we know that the defective gene is still on an autosome, but this time it's carrying a recessive trait. And what do we know about recessive traits? We know that recessive traits do not express themselves if there's a dominant gene present. So in order for somebody to actually have the symptoms of an autosomal recessive disorder, they have to have two recessive genes. If there's a dominant gene present, the dominant one will express itself, the defective recessive will not. So in order to have the disease, there cannot be a dominant gene present. So if you look at the examples at the bottom, The person with the disease has 2 defective recessive genes. What that means, though, is that person had to have gotten a defective gene from both of their parents. So both parents had a defective recessive gene and happened to pass that defective gene on to their child. Now, there are carriers in an autosomal recessive disorder, right? Because if the patient has 1 dominant gene, which is normal, and 1 recessive gene, which is defective, remember the dominant gene is the one that's going to express itself. So this person would not have the symptoms of the disease, but they do still have a defective gene that they could pass on to their children. So that person would be a carrier. In order for somebody to not have the disease and not be a carrier, they have to have 2 dominant normal genes. Does that make sense? Here's an example of an autosomal recessive disorder that I'm sure you've all heard of, and that is sickle cell anemia. The gene that is defective in sickle cell anemia is the gene that has the instructions for how to make hemoglobin. Now, I think sickle cell anemia is really interesting because not only is there only 1 gene out of all the tons and tons of genes that you have, not only is there only 1 gene that has a mistake in it, but that gene only has 1 amino acid coded incorrectly. All the other amino acids are correct, but because of that 1 mistake in the 1 amino acid, now we're making hemoglobin that's defective. And what happens because of that defective hemoglobin is, under certain conditions, it makes the red blood cells become sickle-shaped, as you see in that picture. The problem with having sickle-shaped RBCs is that they clump together and they start forming clots. And so we start seeing people with blood clots that are occluding blood flow to different parts of the body. It causes a lot of pain as well. Now, of course, those sickled RBCs are not considered normal, so the body destroys them. And because we have so many more red blood cells being destroyed than normal, the patient becomes anemic, right? So that's where we get the name sickle cell anemia. Sickle cell anemia is a type of anemia that we consider a hemolytic anemia. So what does hemolytic mean? If you break that word apart, hemo refers to blood or to the hemoglobin in the blood, right? And lysis is a breaking apart, right? So hemolytic anemias, person becomes anemic because we're breaking down our own red blood cells. So people who have 2 defective genes have sickle cell anemia, or sometimes we call it sickle cell disease. The carriers who only have 1 defective gene are said to have sickle cell trait. So I'm sure you know that when we do genetic counseling, in order to more easily visualize a couple's risk of having a child with a single gene disorder or who is a carrier of a single gene disorder, we often diagram it out using Punnett squares. When we do a Punnett square, we're going to draw the boxes and put the parents' genes along the sides, indicating whether they have dominant genes or recessive genes. And then we fill in the boxes with all the possible combinations of those parents' genes that their children could end up with. And we use that to calculate the risk that this couple would have a child with a disease or a child that's a carrier or have a child that has normal genes. So there are some nurses who do get involved in doing genetic counseling, and so I will be asking you to do some Punnett squares on the test. And I find that students often struggle with it, so I try to make you some guidelines for doing Punnett squares. And I think you're actually pretty familiar with it, but this is a good review. Okay, so here I have the steps that we just kind of went through. And I've put together a list of questions that you want to ask yourself when you're doing a Punnett square problem. And we're going to be using these questions as we walk through some examples. Okay, let's look at sex-linked disorders first. Now what I did in the slide is I took the questions that you should be asking yourself as you work through the Punnett square and combined it with the information on sex-linked disorders that we talked about earlier, so that hopefully it'll be easier to answer the questions. So, question number 1. Will you use X's and Y's or upper and lowercase a's when you do the Punnett square? Well, since this is a sex-linked disorder and it does make a difference whether you're male or female, we should use the X's and Y's. Next question. Will you calculate risk out of 2 or 4? Okay, we're going to look at a couple slides that I hope will help you understand why that question's in there. When you look at a Punnett square for a sex-linked disorder, since we're using X's and Y's in the diagram, you can tell which of these children are girls and which are boys, right? The girls are the XX's to the left of the center dividing line, and the boys are the XY's to the right. So, because of this, on the test, when I ask a question about sex-linked disorders, I'm going to say something like, if this couple had a son, what is the risk that their son would have the disease? Or, if this couple had a daughter, what is the risk that their daughter would be a carrier? Now, if you look at the square, you can see that 2 of the possible children are girls and 2 are boys. So when you're calculating risk, you really only need to focus on the 2 children that are the gender that I asked you about. Okay? So if I asked you if this couple had a son, there are 2 boys that they could possibly have, right? So your risk would be either 0 out of 2, 1 out of 2, or 2 out of 2% risk that their son would have the disease. Does that make sense? You really only need to worry about the 2 that I asked you about. You don't need to worry about all 4 of the children. On the other hand, if I'm asking you about an autosomal disorder, we aren't using X's and Y's in these diagrams, right? Now we're using uppercase and lowercase a's, which means you can't tell which of the children are girls and which are the boys. So on the test, if I asked you a question about an autosomal disorder, I would have to say, if this couple had a child, right? And there are 4 possible children that they could have. So now you're calculating risk out of 4 instead of 2, right? So when you're calculating risk, it'll either be 0 out of 4, 1 out of 4, 2 out of 4, 3 out of 4, or 4 out of 4% risk. Does that make sense? So if it's a sex-linked disorder, you're going to calculate risk out of 2. If it's an autosomal disorder, you're going to calculate risk out of 4. All right, let's go back and answer the rest of the questions for sex-linked disorders. Is the defective gene dominant or recessive? Remember we said that sex-linked disorders usually have a recessive inheritance pattern. And you remember that recessive genes only express themselves if there is not a dominant normal gene to override it. So the question about how many defective genes you need in order to have the disease is going to depend on whether you're male or female, right? If you're a male, you only have one X. So if there's a defective gene on that X, there will not be a dominant gene to override it. So in the case of males, they will only need one defective gene to have the disease. Females, though, have 2 X's. And since the defective gene is recessive, if the female only has one defective gene, there is a normal dominant gene to override it. So a female with one defective gene would be a carrier. Does that make sense? Now, it is possible for a female to have two defective X's, but it rarely happens. So we're not even going to talk about that, and I won't do that to you on the test. Okay, so all you need to remember for the sex-linked disorders would be males only need one defective gene to have a sex-linked disorder. Females, if they have one defective gene, they're a carrier. Okay. So then the last question is, what combination of genes represents a person with the disease, or a carrier, or normal genes? I put that question in there because I feel like the thing that students have the most trouble with when they do Punnett squares is figuring out how to represent the genes of the parents. Okay, so here we're just going to quickly review what a male or a female would look like, what their genes would look like if they were a carrier, or if they had the disease, or if they had normal genes. Okay, so the obvious thing would be a person with no defective genes has normal genes, right? So whether it's a XY or an XX, if none of those genes are defective, that's a person without disease, a person who is not a carrier. Okay, so again, we kind of reviewed this. Males only need one defective gene to have the disease. Females, if they have one defective gene, they're a carrier. So let's look at the first example of a couple that I might ask you about on the test. A carrier mom and a dad with normal genes. Okay, so the dad with normal genes is going to have a normal X and Y. The carrier mom is going to have one defective X and one normal X. And, you know, in this diagram, the defective X is the red one. Okay, so if you diagram out all the possible children they could have, okay, you can see which children inherited abnormal genes and which ones did not. So if I asked you if this couple had a son, what is the risk that their son would have the disease? Well, it would be 1 out of 2, so that's 50%, right? Because even though that son only has one defective gene, since he does not have a normal X to override it, he's got the disease. Okay, what if I asked you if this couple had a daughter? Okay, what is the risk that they would have a daughter that's a carrier? 1 out of 2, again, that's 50%, right? Because with the daughters, that daughter does have a normal X to override the defective X. So she does not have the disease. She's a carrier. Make sense? Okay, what about the other example? A mom with normal genes, a dad who has the disease. Okay, now it's the dad that has the red defective X. Okay, so look what happens to the children now. If this couple had a son, what is the risk that their son would have the disease? 0, right? Their sons all have normal genes. If this couple had a daughter, what is the risk that their daughter would be a carrier? 100%, right? 2 out of 2. So, are you seeing how we're calculating risk here? All right, let's look at autosomal dominant disorders next. Will you use X's and Y's or uppercase and lowercase a's? Well, since the defective gene's on an autosome, it doesn't matter whether you're male or female, so we're not going to use the X's and Y's. We're going to use the uppercase and the lowercase a. Next question, will you calculate risk out of 2 or 4? Well, since we can't tell which ones are the girls and the boys anymore, since we're not using X's and Y's, we're going to have to calculate risk out of 4 children. Is the defective gene dominant or recessive? Well, it's right there in the name. Autosomal dominant disorder tells you that the defective gene is dominant, or it's the uppercase A. How many defective genes do you need in order to have the disease in an autosomal dominant disorder? Well, since the defective gene is dominant, you only need 1. right? So are there carriers in an autosomal dominant disorder? No. What combination of genes represents a person with the disease? Well, we said it only takes one defective gene to give you the disease, and the defective gene is dominant, or it's uppercase. So the uppercase and lowercase a is the person with the disease. Person with normal genes would have to not have any dominant genes, right? So they would have the two lowercase a's, the two recessive. So here we're looking at the Punnett square for autosomal dominant disorders. And this is the most common combination of parents you're going to see, where one parent has the autosomal dominant disorder and the other parent has normal genes. So when we diagram out the parent that has the disease, we're going to use one uppercase dominant defective gene and then one lowercase normal gene. Now, theoretically, it is possible for the parent with the disease to have two defective uppercase genes, right? But that situation doesn't happen very often, because if you think about it, in order for somebody to have two defective genes, that means they had to have gotten a defective gene from both their parents, meaning two people with the same autosomal dominant disorder had children, and each passed a defective gene on to their child. That doesn't usually happen because two people with the same autosomal dominant disorder will usually choose not to have children. Because if they both did pass a defective gene on to their child and their child ended up with two defective genes, that child would have a much more severe form of the disorder than a child who only had one defective gene. So most of the time, two people with the same autosomal dominant disorder choose not to have children. So on the test, if I tell you that the parent has an autosomal dominant disorder, I want you to diagram them as having one defective gene and one normal gene. Because again, that's usually what you're going to find out in the real world. Sometimes on the test, people wonder, or they may ask me, so should I use one uppercase and one lowercase a, or should I use two uppercase a's? And if you ask me that, you know what I'm going to tell you? I'm going to tell you, diagram it the way that I told you to in the recording. Okay? So that way is one uppercase, one lowercase. Okay? So remember, we're calculating risk out of 4. So if this couple had a child, what is the risk that their child would have the disease? Okay, well, it's 2 out of 4, which is 50%. And again, are there any carriers? No, remember, there are no carriers in autosomal dominant disorders, because if you even have just one defective gene, you have the disease already. There are no carriers. All right, let's do autosomal recessive disorders now. Are we going to use X's and Y's or uppercase and lowercase a's? Well, since this is an autosomal disorder, again, we're going to use the uppercase and the lowercase a's. Are we going to calculate risk out of 2 or 4? Since we're not using X's and Y's and we can't tell whether the children are girls or boys, we're going to calculate out of 4. Is a defective gene dominant or recessive? Well, again, it says it right in the name, autosomal recessive disorder. That tells you the defective gene is recessive, represented by the lowercase a. Since it's recessive, how many defective genes do you need in order to have the disease? Remember, if the defective gene is recessive, it will only express itself if there is not a normal dominant gene present. So in order for the patient to have the disease, they have to have 2 defective genes. If they only have 1, then they're a carrier. Okay, and we just basically said person with disease has 2 recessive genes. Person with only 1 recessive gene is a carrier. Person with 2 dominant genes, that's going to be a person with normal genes. When you're actually looking at the Punnett squares for autosomal recessive disorders, notice that now there's a lot more different combinations of couples that you could put together. Some have normal genes, some are carriers, some actually have the disease. But as long as you're using the correct combination of upper and lowercase a's to correctly represent the parent's genes, then this should not be a problem. The only thing that you have to be careful of is to remember that you are calculating risk out of 4 instead of 2. So you could end up with either a 0% risk, 25, 50, 75, or 100% risk. So as long as as you make sure that you're calculating out of 4 instead of 2, you should be able to come up with the right answer. Now, we'll do a lot more practice questions in class, so hopefully you'll have this nailed by the time we get to the test. For those of you who want a summary of the information we just covered written out for you in a document, there is one posted in the module for today, class number 2, cell part 2. It's called Help with Punnett Squares. It doesn't really contain any additional information, but it might be easier for you to see it in writing in an organized manner like that. So that's there as a resource for you. And again, we will be doing some practice questions in class. If you have specific questions, make sure you submit those as part of your pre-class exercise. And for those of you who are really interested in single gene disorders, you can look in your textbook, and there is a table that has a lot more examples of sex-linked disorders, autosomal dominant, and autosomal recessive disorders. The other kind of genetic problem we said we would talk about is a chromosome disorder. Now, you remember that chromosomes are made up of a big, long DNA molecule, and that DNA molecule has a whole ton of genes in it. So when we're talking about a problem with a chromosome, we're talking about probably a greater number of kinds of problems than a single gene disorder would have. And the thing is, in a chromosome disorder, all the genes that are present could be coded correctly, and there would still be a problem. We'll talk about a couple of examples of things that could go wrong with a chromosome other than just having the genes coded incorrectly. For example, sometimes during meiosis, when the genes in the chromosomes are being replicated, a piece might break off of a chromosome and get lost. Okay, so if you think about all the genes that are in that piece that got lost, now you can see why there will be a problem, because there will be a whole bunch of genes that are just missing. Another common problem that happens with chromosomes, though, is that during meiosis, when the parent cell is dividing into the two daughter cells, it doesn't divide up the chromosomes equally between the two. You remember we said that when the parent cell splits into two daughter cells, the two daughter cells are supposed to end up with the exact same set of chromosomes as the parent cell had. So the parent cell makes 2 copies of every chromosome, so it can give 1 copy to each of the daughter cells. Sometimes during meiosis, the chromosomes aren't split up equally. Let's take a look at what will happen then. So this is a diagram of what's supposed to happen. You see the parent cell at the top making 2 copies of every chromosome, so that when it divides into the 2 daughter cells, it gives each daughter cell 1 copy of that chromosome. And you remember, this is meiosis, right? So these are germ cells that are replicating. So each germ cell is only supposed to have 1 copy of each chromosome, because that germ cell then goes and combines with another germ cell and becomes a zygote. And each of the germ cells brings 1 copy, so that the zygote ends up with 2 copies of each chromosome, the pairs of chromosomes that you're supposed to have. This process, this is the way it's supposed to happen. We call it disjunction. This is a picture of what happens when the parent cell doesn't divide the chromosomes up equally and instead gives one daughter cell both copies of that chromosome and the other daughter cell gets none. Okay, the problem comes when those daughter cells then get together with other germ cells to make zygotes. Now we're going to end up with the incorrect amount of chromosomes in the zygotes. Okay, so let's look at the daughter cell on the right, the one that did not get any copies of the chromosome. When that daughter cell gets together with another germ cell to form a zygote, notice that that zygote only has one copy of that particular chromosome. And remember, it's supposed to have two, right? Chromosomes are supposed to come in pairs. That zygote only has one copy of that particular chromosome. When that happens, we call it a monosomy because mono means one, right? That zygote only has one copy of that particular chromosome. Now, you don't see too many people with monosomies wandering around because the defects that are associated with monosomies are usually pretty severe, to the point where your book says that they are usually, quote, incompatible with life, meaning the defects are usually so severe that that fetus can't survive and is usually miscarried. All right, let's go back to our diagram to see what happens to the other daughter cell when non-disjunction occurs. Non-disjunction meaning that disjunction did not happen, the parent cell did not divide up the chromosomes equally. So now we have the situation where the daughter cells don't have the right number of chromosomes. So we talked about what happened to the daughter cell that had no copies. Let's talk about what happens to the daughter cell that gets both copies. Okay? When that daughter cell with two copies of the chromosome gets together with another germ cell and makes a zygote, how many copies of that chromosome does the zygote have now? It has 3, right? How many is it supposed to have? It's supposed to have 2, but it has 3. When that happens, we call it a trisomy because tri means 3. And there actually are quite a lot of trisomies out there. There's trisomy 18, trisomy 13, trisomy 22, with the number referring to which chromosome there is an extra copy of. But probably the most common one is trisomy 21, otherwise known as Down syndrome. So this is the karyotype of someone with Down syndrome or trisomy 21. You'll notice that all the chromosomes are paired up except for the 21st chromosome. There are actually 3 copies of that. You can see it in the lower left corner. Now, this is actually not the karyotype of that child that's pictured because you notice the child is a girl, but the karyotype has an X and a Y. So that's actually the karyotype of a male. But I wanted to show you this because here's your chance to see that the Y chromosome is a lot smaller than the X. Remember we talked about that earlier. All right, so because of that extra chromosome, children with Down syndrome have certain kinds of physical and mental abnormalities. There's usually some characteristic facial features. There's usually some degree of mental retardation present, and there may be some other genetic defects, congenital heart problems, GI malformations, all those kinds of things associated with that extra chromosome. So again, you can see how we're not talking about one specific problem anymore like we see in sickle cell anemia or hemophilia or some of the single gene disorders, but we're talking about a whole constellation of problems that occur because of a problem with a chromosome. As far as assessment findings and interventions for chromosomal disorders, we're pretty much in the same boat as we were with the single gene disorders, where we can't make any blanket statements about assessment findings that are common to all chromosomal disorders, because of course the problems that occur with a chromosomal disorder is going to depend on which chromosome has the problem, right? And since interventions are geared towards treating the symptoms, and the symptoms are different depending on which chromosome you're talking about, there really is not any one blanket statement we can make that would be true for how we treat all chromosome disorders. It's going to depend on which chromosome is involved, what kinds of problems it causes. But pretty much we are left with trying to treat the symptoms and give the patient as good a quality of life as we can, because at this point we do not know how to go and fix the chromosome problem, and so we don't have that many treatment options. You'll probably study a lot more about these chromosomal disorders in peds, I'm thinking, because these are problems that babies are born with, and that's the time of life where the families are learning how to care for them and to facilitate their best development. So you'll revisit this when you get into peds. All right, that's it for now. Remember to submit your questions. I'll try and answer them in class. I will see you then. Bye.